What is cryopreservation freezing? Imagine stopping time for living tissues or cells, keeping them perfectly preserved for years. That's what cryopreservation freezing is all about. It's a way to use super cold temperatures to pause biological activity. When you lower the temperature to between -80 degrees Celsius and -196 degrees Celsius, metabolic processes inside cells nearly stop. This is often done with liquid nitrogen, which is extremely cold and helps reach these temperatures quickly. From a thermodynamics point of view, freezing involves phase changes, mainly water inside and outside cells turning into ice. When water freezes, it releases heat called latent heat of fusion. Managing this heat releases is key because if ice crystals form too quickly or in the wrong way, they can damage cell structures. Ice crystals can puncture cell membranes or cause osmotic stress, which harms the cells. To avoid this damage, scientists add special substances called cryoprotective agents. These chemicals, like glycerol or dimethyl sulfoxide, penetrate cells and lower the freezing point of water. They help prevent the formation of large ice crystals inside cells, which can be deadly. The cooling rate is also carefully controlled. Cooling too fast can cause inside cell ice crystals, but cooling too slow can lead to harmful concentration of solutes outside the cells. The goal is to find a balance, allowing water to leave cells gradually before freezing, reducing ice damage. In practice, devices that control temperature are used to gradually lower the temperature in stages. Sometimes, ice formation is induced at specific points to avoid supercooling, which is when water cools below its freezing point without turning into ice. Alternatively, rapid cooling methods like vitrification freeze water into a glass-like state without forming damaging ice crystals. This is often done by plunging samples into liquid nitrogen. Cryopreservation is widely used in biology and medicine. It helps store cell lines, tissues, and reproductive cells for future use. It supports organ transplants, regenerative medicine, and even the preservation of endangered species' genetic material. Understanding how phase changes and thermodynamic principles work is essential for making sure these biological materials survive the freezing and thawing processes intact.